of Malaya. University of Malaya is located in Kuala Lumpur, Malaysia, and right now it is top 100 ranked in the QS university rankings, and actually it's ranking 65 in the world. And Profit is the one of the founder members and the current secretary of the Consortium of Southeast Asian Seaweed Taxonomy, or SEASTAX, and currently she has more than 100 publications, international index, and probably 180 publications. And with the she has also initiated lots of other projects such as GCRF, Global Seaweed Star Program, where problem-based multidisciplinary approach adopted to address several main issues affecting the seaweed cultivation and processing industries. And Professor Lim already has lots of experience in developing DNA biomarkers to identify algae, well, microalgae mostly. But I think we can learn a lot, because in order to us to evaluate the potential of various natural products, one of the most important things is we have to determine the exact species, and for doing so, we cannot rely on morphological identifications only, but we have to also consider DNA molecular identification. So today we will learn a lot from Prof. Lim on probably the how do we, if we want to do DNA molecular identification of various natural products. So Prof. Lim, the lecture time is around 60 to 90 minutes, but I will give the freedom to you to adjust the time.
Okay.
So Prof. Lim, the time is yours. You're welcome to start the lecture.
Okay.
Thank you very much.
Thank you, Dr. Eka. Let me share the slide first. Okay. Okay, a very good afternoon to all of you. Okay, so I will be able to handle the Q&A question if you have at the end of my lecture. Okay, so firstly, I would like to thank Dr. Eka for inviting me to share with you the lecture on the importance of correct identification of seaweed for pharmaceuticals application. So in general, I will try to share how important correlate for pharmaceutical application, but I would like to highlight that actually the correct identification is very important, irregardless for whatever application that you have in mind. It can be for aquaculture, it can be for nutraceuticals or nutrient, whatever is it. So let us go through my lecture. Okay. So if we talk about ocean, okay, we know that blue economy is actually one of the primary or important sector that our government is putting a lot of emphasis and knowing and realize it's important. Because one of the things is, if we talk about ocean, more than 70% of the world are comprised of ocean, and we have more than 80% of the world living organism are living in the aquatic, and it is estimated 40,000 species are found in ocean, ranging from microorganisms, sea grasses, algae, coral, and animals. So when we have a large pool of marine organisms, it means that there is potential for us to look into some of the biological active compound. Okay, so and why ocean? We say ocean is a promising source for us to obtain various kind of compound. This is because the marine organisms are living in a condition where they are exposed to different conditions, okay? In compared to terrestrial, they are exposed to physical, chemical, and hydrological parameters such as fluctuation of temperature, light intensity, salinity, and pressure. Any organism, when they are exposed to different kind of parameter environmental they're growing, they will they themselves will come up with strategy so that they will continue survive. So in that case, because of their strategy to overcome that, they also will provide a great bioresources for secondary metabolites such as enzyme and biopolymer. Okay. However, ocean is considered one of the area that is underutilized, underexplored, and under-valorized resources. Thus, it gives a huge potential. So, and now we are living in the era that marine biotechnology has advanced in the stage that there are many available tools that actually make or accelerated the process of discovering new compound or new application. So progress in methods such as genomic, proteomics, metabolomics, or transgenics, fermentation, gene therapy, bioprocesses technique, bioreactor, and bioinformatics has actually helped to enhance or to accelerate the potential, okay, or speed up the process for us to discover new bioactive compound, okay, and this have give a promising breakthrough for aquaculture, microbiology, metagenomics, nutraceuticals, pharmaceuticals, cosmeceutical, biomaterial, biomineralization, biofouling, and bioenergy. There are many areas of the application as well as research actually supported with the new advancement of tool in marine biotechnology. Okay, if we talk about what are the potential products from marine biotechnology, they are huge. Okay, so one of it people that are focusing of discovering new compound is they look into the biomolecule, mainly are secondary, which is metabolites, enzyme, and biopolymer, which are variable resources for development of product. Okay, so biomolecule produced in organism can be used for mediates the chemical communication between organism, marine organism, or any organism. Actually, they do communicate, but the way of they communicate a lot is different from us as human beings that we talk, but for them, it is through a chemical communication. And at the same time, all the organism also, they have a self-protection skill so that they can survive to various environmental conditions, or they can protect them by having weapon for catching prey or for protection against predator, pathogen, extreme weather, or harmful UV skill. All the adaptation, they produce a different type of compound. So with that, we are able to explore, and hopefully we can harness some of the bioactive compound that's useful for further application. Okay, so based on Newman and Craig in 2020, it found that there are more than 50% of the medicine are derived from originated natural compound, and it is especially higher for anti-cancer and antimicrobial treatment. So, and we know that at the moment, because of health conscience, people are actually moving towards, when we talk about looking for new resources of medicine rather than using a chemicals derived compound, people are looking for the natural resources which believe it is less harmful and for the people that are taking it. Okay. In addition to this, okay, other property and function of marine organism can also beneficial and of interest to various industry. So they can be used for example, removal and degradation of individual chemical compounds or organic matter. In this case, we are talking about bioremediation approach, okay? And then seaweed has been also become one of the popular resources because one of it is seaweed are rich in carrageenan or other types of polysaccharide. which they already been used as a food additives and cosmetic ingredients since the 1930s. But with the application of marine biotechnology, it actually helped to intensify further application of this marine organism as well as their secondary metabolites. Okay, this is the table showing you some of the examples of the uses of the product derived from marine. Okay, so I won't go through every one of them, but I would like to highlight as regard for microalgae, especially, which is mainly, which is, or it is known as seaweeds. They are used mainly as a food, feed, medicine, cosmetic, nutraceutical, biofertilizer, soil conditioner, biomaterial, bioremediation, and energy. So you can name that there are a lot of applications when we talk about seaweeds. Okay, so seaweed in short, okay, we know they are known as microalgae. They are multicellular and photosynthetic organisms that use sunlight and nutrient present in the seawater for photosynthesis and produce food. Okay, the main group are categorized according to their pigment. So in general, we will say seaweeds compound of three main groups, which is red seaweeds, or we call them rhodophyta, green seaweed, chlorophyta, and brown seaweed, or chlorophyta. Okay, and they are widely used for food, medicine, pharmaceutical, and nutraceuticals. Okay, this is the simplified representation of a marine biotechnology product. Okay, this is a pipeline, that a step that involved if you want to produce any product for applications, for commercial application. And it have to start off with the very fundamental, which is the basic research involved on the ocean observation, biodiversity study, sequencing, profiling, bioassay. So what I would like to highlight is before you can reach out to applied research, industrial scale up, and commercial application, most of the important thing is we must know the identification on the organism that we work on. Because you can tell people, hey, I have this seaweed that is very good. And then the next thing people will ask you, what are the species? You cannot tell them. They look like green, they look like brown or red, but you have to tell them exactly what are the names. Okay, so that is where it's lead to, I'm going to share with you the importance of correct identification for seaweeds for further application. So today I will mainly share with you on the use of molecular tool in identification. Okay, so before we go there, if we know that in terms of the value of dollar and cents on the application, okay, this is a pyramid. If you talk about the usage, okay, in terms of biomass, more is energy, food, feed, agronomy, and bioremediation, which needs not high-tech or shorter times when you want to develop. But as you reach to higher level medicine, in terms of volume, number is less, but in terms of the value, the return of investment, it will be higher if you talk about medicines. Okay, so now let us look into why it's so important of using genomic approach or DNA for correct identification. So I give you some definitions. Okay, when we talk about identification using DNA, we will say that it's molecular taxonomy, because it is important. It's mainly seaweed are highly phenotypic, ecotypic, and plastic seaweed. Okay, let me define. Phenotypic means that they are the appearance, which is the look. Ecotypic, it can be changed according to the ecological condition they are grown. And plasticity means they will change according to the environment. So for seaweed, if you grow in location A, when you move to location B, there is a possibility that they will change color because is their nature. They are plastic, and then they will change to according to their environment. So in this case, how do you sure what is the correct species? That is why when we talk about seaweed identification, using morphological, it is very difficult to correctly identify. But I won't say that it's not possible unless you are trained and you have the experience and you also regular go to the place and you have already confirmed the species identification before that using molecular marker that will help you what is the diagnostic character referring based on DNA data that you will know the species, okay? And correct identification is especially important for seaweed cultivation. It started with seaweed cultivation, but I believe the application is very important as now seaweeds have gained its importance, not only for aquaculture, but also for discovering a new bioactive compound that potentially to be used for pharmaceuticals and medicine purposes. Because correct identification is the best way to predict the nature and property of its component. Okay. Beside that, there are also studies have been carried out and they have identified a specific marker that you actually can link them with growth or disease resistant, high yield or special compound. Okay, so let us look into the species identification selection. Okay, species identification of seaweed is rather challenging because as just now I mentioned, seaweed are morphological plastic. Individual of the same species may look different from another one and vice versa. And beside that, study have shown that when you culture them, because seaweeds have a very unique feature, they can reproduce either sexually or asexually. In this case, the clones, which is without sexual, their variation is also common among the cultivar. And this is evident based on the Kappa ficus and eucuma that we culture in the water, which I'm going to share with you later. Beside that, sometimes the species that you collect, okay, from the natural population, they can be damaged or deformed and difficult to identify based on the morphological feature, okay? However, but with the available of DNA technology, it helps us to do selection, in which selection can be done more reliably based on intrinsic property of the seaweed instead of extrinsic one like morphology. Because DNA, you will test on the actual composition in that organism, but if you test on the morphology, it will change based on the growing condition, okay? And when we talk about identification using DNA, okay, we are saying that this is done by identifying or analyzing the specific DNA region of the seaweed, which is known as DNA or molecular marker. Okay. Let us look into genomic tools for identification and selection. So, strain selection and taxonomy are often linked to each other and may utilize the same technology or techniques. Okay, some of the example of the conventional method that you use for strain identification and selection are random amplified polymorphic DNA technique, or in short, we call RAPD or RAPID, or restriction fragment length polymorphism, RFLP, and amplified fragment length polymorphism, AFLP, and the newer technology that involve linking digital information to economic trade, such as DNA sequencing, microsatellites, single nucleotide polymorphism, as well as marker-assisted selection. Marker-assisted selection is normally you will link them with a certain trait, like high growth rates, high yield, or disease resistant. Okay, so this is bad. Okay, let us look into the overview on the timeline as regards about the progress of the use of DNA marker or molecular taxonomy. Okay, the identification, if you look into in the old days, from late 1960 to 70, in order to identify a correct seaweeds or correct species of seaweeds, in the old day, they used protein electrophoresis, which apply to isozyme and allozyme. And then in the late 1970 to 80, people are using RFLP analysis on the mitochondria DNA. It can be mitochondria DNA, it can be genomic DNA. Mid-late 1980s, DNA fingerprint, such as using RAPID, okay, random amplified polymorphic DNA. And then since 1990 up to present, DNA sequencing, AFLP, single strand confirmation, or SSCP, have been used, okay? And 1997 until present, people are looking into using express sequence text, DNA sequencing, and we are, especially nowadays, we are no longer talking about single gene for identification, but you use a few DNA marker. And then we are also even looking into phylogenomics. In this case, we are using the mitochondria as well as plastic organelle used for differentiation. Okay, civic taxonomy. Okay, identification and species delineation is problematic due to lack of distinguished morphological characteristic and plasticity of the seaweed. Because when we look into seaweeds, the look are very simple, and then among the different species, sometimes you have very little of distinguished feature that help you to identify. And this is further complicated when they are restricted with the plasticity, means they somehow will change if they're growing in different conditions. Okay, but the breakthrough in biochemistry and molecular biology had caused a new development for algae systematic. Okay, so we will look into that. Okay, so let me share with you. Phycologists start employing molecular approach for seaweeds taxonomies in the late 80s. Okay, probably many of you are still not born yet. Okay, for example, for green algae or green seaweeds, it started by Olson et al. In 1987. This is our first few study. And then for brown seaweed, it started by Lin et al. 1983, 96, and Fain et al. And then for red seaweed, is Goff and Coleman. So these are the first few studies that actually lay the foundation. Subsequently, many people are looking into using the DNA molecular marker to identify the seaweeds. Why taxonomy? Because taxonomy study is important, so they make sure correct seaweeds are identified and recognized in order to fully utilize seaweed resources and enhance the development of both phycoculture as well as the seaweed product processing industry. For example, strain selection to find a fast-growing, good-grade algae is very important before embarking on mariculture, because variable quality of algae obtained from Gastrularia may present a problem. Because, for example, if you are the farmer, you decided to start a farming. So let's say for Indonesia, you culture a lot of Kappa ficus, right? So, and then for the Kappa ficus, you have two types of the seaweeds in that group. You have Kappa ficus and Eucheuma. Kappa ficus produce Kappa carrageenan, which have higher value. And then another one is Eucheuma. They produce carrageenan that is less lower in terms of price. But if you don't know, you just happen to choose to take Ukima, which is giving you iota carrageenan, is much lower price. So you need to make sure you identify you culture the correct species before you started your culture. So I'm going to share with you some of the technique as regards about the methods for molecular taxonomy, which is DNA based. Okay. We are going to look into single copy DNA hybridization, restriction, fragment length, random amplified APFE, and sequencing. Okay. Okay. Firstly is if we talk about the RFLP, okay, estimate DNA sequence divergence by detecting pattern variation in DNA fragment length caused by changes in the bases. For example, substitution, deletion, and insertion of nucleotide across the entire genome using restriction endonuclease. So in this case, the enzyme that you are using, which is restriction endonucleases, it will identify and give a different pattern should there in the fragment of the DNA that you analyze, they have events such as like substitution, deletion, or insertion. So this method has helped phycologists to explore phylogenetic at the population, species, and genetic level in various algae species. This method is one of the earliest methods that has been used. But however, this method overall, if your sample size is small, it's easy to handle. But if you have large sample, it is quite time consuming. And also this is expensive. And most of the challenging part is actually this technique will require a large quantity of high quality DNA. And if you want to compare from laboratory to laboratory, it's difficult. So you can't like today, probably you are at Umrang and then another one person in University of in Sulawesi that would like to try to repeat. But the problem is it's difficult for you to compare the result of yours with another laboratory that carry out. Unless you have standardize all the method that you use, then it is possible, but it is time consuming and need a high skill in terms of good technique for you to pick up the skill. Okay, this is some example of the study have been carried out. As you can see, most of the references, okay, is earlier time. So is as I mentioned, this is one of the new first few techniques when the molecular taxonomy come in. So it's being carried out for brown seaweed as well as red seaweed. For example, like Mycocystis costara laminaria, this is mainly for the temperate species. As for Rhodophyta, you see a lot on Gasseralia. And then if you talk about Porphyra, this is temperate species. Okay, so if you talk about the identification using of molecular tool, it started more in those countries in the West or temperate species, but now we are using it in our study that molecular tool is important in order for us to help us to identify. Okay. Another second method is rapid. Okay. Random amplified polymorphic DNA technique. So this technique is heavily depend on the polymerase chain reaction of PCR. In this case, you will, based on the PCR, you actually differentially amplify any anomalous region of DNA using a single short oligonucleotide primer. In normal case, when you do a PCR application, you will need a primer that amplified both direction. But in this case, for RAPD, the primer that you use are single, one only, and also very short. So in this case, by you having a short primer, it will increase the possibility of this primer to anneal to the template and will amplify. Okay, so but because of that, the probability of randomly amplified, it also causing a problem with in terms of reproducibility in study. Many publications says that the problem with rapid is easy to use, it's cheap, it's fast, but there are some studies showing that they have problem of reproducibility. For myself, personally, we have used these rapids in the early day, and we found that you can overcome the problem of reproducibility if you keep all your parameters experiment constant. You do not change, such as you use the same parameter for your PCR amplification. Even your tag polymerase for amplification must be the same brand. You must use the same thermal cycle, everything same, then you can get a reproducible. But if you have such a stringency of having everything have to be keep constant and not change, then you have the problem if you want to compare from laboratory to laboratory. As regards about the usage, it can be applied to detect differences between related species, strain, and cultivar of many organisms. And it have been shown that useful in the taxonomy and classification of red and brown seaweed at the genus level as well as species level. Okay, so RAPD, as I mentioned just now, is relatively fast, easy to perform, inexpensive, it is not costly, and require no pure knowledge of the genome to be investigated. Normally, there are already random primer that is shown in the market. So normally you will screen through many sets of the primer, and then you will, after that, you will finalize or shortlisted those primer that can give amplification. And beside that, the advantage that it have compared to the RFLP technique that we mentioned just now is the amount of DNA that you require is very little, and because we are using PCR technique to amplify that. Okay, the reaction product can be resolved and documented easily and quickly using gel electrophoresis and photography. So in terms of that, in terms of the investment of facility that your laboratory need to have is relatively easy. You don't need to have a DNA sequencer, but you will check the profile after the amplification by subject to DNA at the gel electrophoresis. Okay, these are some example of the RAPD technique being used for seaweeds. Okay, it has been studied used for Phaik, okay, the brown seaweeds from Sargassum, Hesychia, and Laminaria, and then Rhodophyta of Gelidium, Porphyra, Gasseralia, Digynia, and Postilla. So there are quite a number of studies have been carried out, and this is, you can see, is that mainly in the 1990s more. So as subsequently it come to now, currently less people are using. The most reason that why they are not using RAPD is because it does not allow comparison from one laboratory to the other laboratory. Another thing is due to also there is a reproducibility problem. Okay, but reproducibility can be overcome if you keep all the parameters constant. Okay, another molecular techniques that I would like to share is microsatellite. Okay, what is microsatellite? Microsatellite is a short repeat motif, usually 1 to 6 base pair in the region of eukaryotic organism genome. The repeat can be perfectly repeated tandemly or interrupted by several non-lipid nucleotide or compound lipid. Okay. So normally you will have like, probably you have AC, 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 or AGC, AGC, AGC. It can be repeat a few times. So people are using that as a differentiation, and a lot of time microsatellite is useful, especially to use for strain identification. Species can be used, but depends on the markers that you can find. Okay, microsatellite are using associated with high level of frequency of length polymorphism. It is also referred as simple sequence repeats, or SSRS, and it can be found both in coding and non-coding regions of DNA sequences of higher organisms. Okay, so nucleosatellite loci have been developed for brown, for example, the fucoid species, the Icophyllum nodosum, and the Cap Raminala digitata. And then for Gasseriella gassilis and Gasseriella species, okay, that was actually published by my student. And we look into that, okay, as well. Okay, and one of the latest technology since 1990 and up to date is still widely viewed is DNA sequencing. So when we talk about sequencing, is actually we want to know the exact nucleotide sequence in a fragment of DNA. It can be a genes or franking region or non-coding region. Okay, and one of the advantage of we talk about DNA sequences is it is possible for us to compare one laboratory to another laboratory. So it means that what you are doing a research, probably you only need to focus on a few samples from your lab, from the your laboratory, and then you want to know whether what species are there. You actually can link that or search from gene bank or any public DNA databases and download them and compare the sequences. Then by doing the phylogenetic analysis, you are able to determine this what species that you are working on. Okay, so with the ability of sequence data in gene bank, it makes the identification of the unknown species possible by comparing the sequence with the existing data in gene bank. So you can see that I share with you about RFLP, RAPD, microsatellite. However, the previous three techniques is hard for you to compare, but sequencing allows you to compare from one laboratory to laboratory, and then you can refer of the sequence data in gene bank. So that's why this technique is popularized and is still using today. So that's why if you ask me, Prof. Lim, what would the technique that you will recommend us to do if we want to identify the seaweed that we are working on? I would say you go for it, you go for DNA sequencing, okay? But when we talk about sequencing, it's not just simple. The next thing is you want to ask, when you want to identify for taxonomy purposes, there's no need for you to do the whole genome, right? If you do a whole genome, it's very costly. And also you have problem when you come to analysis such a huge data. So normally you will focus on specific region, coding or non-coding region. So I'm going to share with you some of the region of amplifications that you will target before you subjected for sequencing that we are commonly using for seaweeds, okay? So some of the common regions that we are using is in the early day is focused on nuclear genome, which is on ribosomal DNA, which is the repeat in the eukaryote comprised of like 16S light DNA, which is 18S RNA, or large subunit can be S rDNA, as well as 5' rDNA, and also IDS 1 and 2, the two internal transcript spacer, and the external transcript spacer, the intergenic spacer. So basically, these are the arrangement of a ribosomal DNA, you have 18S, frank with ITS1, 2, ITS1, ITS1 and 2, they are non-coding regions, okay? But for 18S, 5'S, 28S, they are coding regions. So normally for coding region, when you come to differentiation, normally is less compared to non-coding region. But for ITS, sometimes you have complication in terms of analysis because ITS tend to form a secondary structure. So sometimes you have to be careful when you come to do the analysis and alignment. Okay, and then another regions that you can, for your consideration, is you can look into the plasmic or chloroplast genome. Okay, and some of the marker that is widely used for CVID are RBCL. In fact, this is one of the markers that is widely used for brown CVID, red CVID, as well as also green CVID. Besides that, we also have PSA, PSAA, PSBA, and PSBAB. So this are the sum of the marker. So another thing is another region another regions is the rubisco. Okay, when we talk about the rubisco or the ribosylase bisphosphate carboxylase oxygenase, which is the RBCL, which is chloroplast encoded gene. It is discovered in 1948, and it is located in the stroma of chloroplast. It catalyzed the first irreversible step in photosynthetic carbon metabolism. The carboxylation of ribulose 1,5-biphosphate by gaseous carbon dioxide. So, and there are two forms. Okay, one is the two structure form of rubisco based on subunit composition. One is the large subunit, one is small subunit. So, but if based on the, in terms of the applications, actually RBCL is more widely used compared to RBCS. Okay, so the chloroplast encoded RBCL gene and also the rubisco spacer region have been employed recently to infer the phylogenetic relationship among algae, especially for generic and species comparison in the Phaeophyceae and Rhodophyceae in the red and brown. Okay, the RBCL gene is known to have a more even distribution of mutational changes along its length than they observed in 18S rDNA. Okay, 18S rDNA was used in the early stage. Okay, they are from nuclear genome, but subsequently they are not so popular is because they do not give much information because they are very conserved. When we say conserved means the sequence of among different species, they are quite similar. So people are looking into, and genes, for example, like RBCL, you have a distribution of mutational changes along the region that you use. Okay, but there are also some authors concluded that rubisco spacer is too conservative to resolve differences between the population. So in short, the different type of regions or genes that you decided to use is actually depend on the group of CVIDs that you want to study, as well as also what level that you want to look into. Do you want to solve or identify based on species, or you are interested in a family level or higher level? So these are the things that normally we will start to ask ourselves before we decided. Okay, we say yes, we want to do DNA sequencing. Then the next question we'll ask, what are the genes, what are the markers that we want to use? Okay, of course, now also mitochondria region is another one that people are using a lot. And mitochondria regions, for example, COX-2,3 spacer is using a lot, especially for the Kappa ficus and Eucheuma species. Then this marker shown to be variable within species and even within population, and have potential to be used as marker for population and demographic study on red algae. It has been used for Gassirella species as well as Bostricia. Bostricia is the red seaweed that you commonly found in the mangrove area. Okay, and another one, genes that is very popular is COX-1, because people think that this is a barcoding marker that is actually widely used for fish, for animals, and it is actually also found, it is quite very useful even for red seaweed, especially in our group. We discovered it's useful for the study of eucheumatoids, the Kappa ficus and Eucheuma, especially up to the strain level. Okay, and then if we talk about, in order to increase or to provide a more stable or reliable result, people are looking into multiple genes or multiple regions. Sometimes people like to compare that in order to be reliable, you should use gene that is conserved, for example, from nuclear region, then moderate conserved, like plastic, or more variable of using mitochondria. But which region or which marker to use, you have to always look into the data bank. What are the genes that are already available so that you can do comparison? And also you have to refer to the literature. Okay, so for example, identification of Atlantic porphyra using small unit of ribosomal RNA nucleus and plastic. So in this case, they combine the genes from the nuclear region as well as plastic, or you can say it's chloroplast. And then for relationship of ceramian sinicola complex, so in this case, the species, they are not sure whether all these complex belong to the same species or not. So in this case, they use the plastic encoding region, RBCL and Rubusco, as well as nuclear encoded gene small unit of ribosomal DNA. Okay, and also mitochondria, the multi-region, they also use for phylogeography. In this case, they want to see like the same species or the complex on different location. Are they saying the similar genetic composition or not? So in this case, they are using large subunit ribosomal RNA nucleus as well as Cox-truth spacer from the mitochondrial region as well as the rubicular spacer. So in this case, they combine all the three markers from the three main organelle of seaweeds, which is nucleus, mitochondria, as well as plastic. So, and then it can be also multiple regions from the same genome. For example, Griffithia, they use 3 genes from the plastic genome, which is RBCL, PSAA, and PSB, PSABH. Then phylogeny of Phytomyophyceae, which is brown seaweed, based upon a 5-gene molecular phylogeny, nuclear encoded SSU and plastic encoded RBCL. So this is the study that been carried out. So another method is the single strand conformation polymorphism. So this is the method that you used for detection of single point mutation. It is rather simple and also very fast. Okay. So for example, SSCP analysis on the Rubusco spacer was used to detect the variation on red seaweed such as Bostricia and Cargolosa. So these 2 genus are common in the mangrove area. So SSCP is a simple and rapid technique for detection of plastic variation and haplotype between the population of red algae, and has high potential to be brown algae as well. Normally, SSCP you are using for the same species, but you want to see the differences, whether they are the same genotype or haplotype. So now let me share with you some of our research in my laboratory, how we have used this different approach to differentiate the species. Okay. So RAPD was used in our laboratory in the old day, pure before DNA sequencing being popularized. Okay. So in this case, we are using molecular taxonomy using RFLP on Gasseralia. So in this case, we found that it is very useful for taxonomy study to differentiate the Gassera salicornia, Gassera edulis, and Changai. We know that when we talk about Gasseralia, a lot of time it's very hard to differentiate because they are all bushy and also the tail line, and they have very few distinguished molecular feature. At the same time, we also used to study the population study on Gassera Changai, collected from different locality of Peninsula Malaysia and South Thailand. And then subsequently, my PhD was partially on that, in which we are using RAFP to differentiate of the closely related species of Gassera salicornia. Gassera salicornia was merged, okay, by Professor Chia Pangmei from China. She's a very famous taxonomy for Gasseralia, which she combined the different species of salicornia, which is Gassera caliculata, also known as Carasa, Cacalia, and minor, into one species, which is salicornia. Okay, because she found that it's very confusing and difficult to find the boundary or distinguished feature to differentiate the four species, and their features are always overlapping and cause confusion. Because of that, she merged them, and then my study is to use RAPD to see whether the combination is valid, or are these four species similar or same. So in this case, we collected the Gassera canaculata, or known as Carasa, Cacalia, and Gassera salicornia were collected from various locations in Peninsula Malaysia, and their taxonomic position were examined using RAPD. Okay. So in this case, I actually screen initial stage. I screen about 100 primer, and then out of the 100 primer, we found that 9 primer are able to give a polymorphic or differentiation of in terms of the differences in terms of DNA fingerprint. Okay. So in this case, the fingerprint from the 5 primer were analyzed, and cluster analysis were carried out based on the metric similarity of the RAPD marker, so that the specimen were grouped according to the location, as well as also to the type of species. So let us look into that. So you can see that based on marker, group 1A is this MB is from Morit, one location, salicornia, and then we have Gassera canaculata from Tanjung Tuan is differentiated, and then Gassera canaculata from PJ is also separated despite they are same species, and also salicornia. Salicornia B and A is actually they are differences in terms of morphology as well, and then canaculata from different location, they are differentiated. So we found that actually RAPD marker have the ability to differentiate the type species as well as also the location for population study. So the RA result shows that Gassera salicornia, Gassera canaculata, and Cacalia are different genetically, okay, based on RAPD. But the thing is, if you would ask me, what is the result if you carry out using DNA sequencing? So if we carry out the DNA sequencing, there are some changes, there are differences, but I would still consider them, they are similar species because differentiation is very small. So in this case, I would say is that RAPD is useful to identify up to the strain level. Okay. Another study is using RAPD marker to differentiating male from female as sporophyretic thalli of Gassera changai. Do you know that seaweed have sex? They are female plant, male plant, as well as sporophyte, which is 2N. So they can be male or female, which is haploids, and then sporophyte thalli, which is 2N. But the thing is, if I take a seaweed to you and ask you to identify and tell you, can you tell me this is a male seaweed or female, or they are sporophyte stage? You can't tell me until you have to examine them under microscope. But if you want to examine them under microscope, you won't be able to recognize male and female plants unless they are fully mature. So in that case, it will be useful if we have some marker, molecular marker that help us to differentiate from the differentiated male and female sporophyte thalli. So in this study, it was carried out in my laboratory. This is carried out by my student. So we managed to find a marker that was present only in male plant but absent in female and tetrasporophyte, which is the case of the marker of 716. Okay, so if you are interested, I'm happy to share with you the publication. But the thing is, identification of male is important because male is actually, in terms of ratio of plant that you found in the field, normally male plant is less compared to female and the tetrasporophyte plant. Okay. So another group of the seaweeds that I would like to share with you of using is on Kappa ficus and Eucuma. Okay. In the early day, we use microsatellite. But the thing is, when you do the RAPD and microsatellites, it's hard for you to carry out comparison. So if we want to do a larger scale comparison with your data, with whatever is available in GenBank, then you have to do DNA sequencing. So with that, we have shifted our research and we identify our seaweed using DNA sequencing. Okay. So I would like to share with you on the phylogenetic and taxonomy study of Kappa ficus and Eucuma in Malaysia as well as Southeast Asia. So for Southeast Asia, we are happy that colleagues from Mataram University, so Prof. Sunapi as well as Dr. Aloh, and in fact, Dr. Eka have done that for his master. Okay, and then we have collaborator from Vietnam as well as Dr. Anisha from the Philippines, where we look into that. So you can see that if you talk about Kappa ficus and Eucuma, you go to the field, you can see all kinds of the plant and structure, and people are giving them different local name. So our first study is we want to know the different of shape, different local name, what are the species do they belong to? So that's where we started, okay? And we know that Kappa ficus and Eucuma, economically, they are very important, in which they produce carrageenan in the world and fetch a lot in terms of economically. And Indonesia being the largest producer, and it have actually bring a large revenue for the country as well as for the farmer. But this group of species, they have a problem of morphological plasticity.
Sama ini. Saya tulisan kita, bro.
Okay. So they have a problem of morphological plasticity. So this is, for example, the local name that is given for Malaysia. So, for example, these two, they look like a bouquet of flower. So the farmer call them based on the color. If green, they call it green flower. Yellow, you call it yellow flower. I think in Indonesia, you call it sako. Okay. And then we have another group of the seaweeds that, because at the end of the thallus, okay, it look like a hands, okay, tangan-tangan. So they call it tangan-tangan, like a hands. And then tambalang is actually named after a large farmer from Philippines, because Malaysia, the cultivation site is at Semporna area, which is neighboring to Philippines. So we have a lot of influence in terms of using the name. So they call it tambalang green based on the color, tambalang brown by color. And then for tambalang variety that's very huge, can grow very big, they call it giant because it's like giant. And then aring-aring is very slim compared in terms of the thalli. And then we have another variety that we call boya. Why boya? Because of the appearance, it looked like the crocodile scale. So we call it. And then spinosum, we know that it is due to this existing of spine throughout the thalli, which is people call it spinosum. Spinosum come from the word spine. Okay. So with that, we carry out, and then the species, the variety that we call aring-aring turned out to be a new species based on molecular data, which we name it Ficus malaysianus, means the region referring to Malaysia, Philippines, as well as Indonesia, named after the region that it is found. Okay. So this is the example of how we carry out. So in this case, we carry out the molecular marker, DNA sequencing of COX-2 spacer, COX-1, COX-2, as well as RBCL. And based on all these 4 markers, it shows that they are differentiated out and it doesn't share with any existing species. So thus we name it. We name a new species, which is Ficus malaysianus. So these are the various study that I'm not going to discuss in detail, but we study Malaysia region as well as also Southeast region as a whole. Okay. Another group that we are looking into is Jellydialis. Okay, Jellydialis is one of the group of the seaweeds that actually give a source, the best source of high quality of agar and agarose. Okay, agar is commonly in our region, we use Gasseralia. But in terms of, you want to talk about good, really good quality agar, is actually Jellydialis. Primarily, some of the species that is important is Thelocladella as well as Jellydium species. But in our regions, I think for Malaysia, we do not have Jellydium, but we have Thelocladella. Besides that, this species also actually can be used to produce high quality of pulp, which can eventually produce a high quality paper due to the hollow structures, the presence of the fiber inside the seaweed. So our project is actually wanting to find the suitable strain or species of the Jellydialis that we can potentially culture in Malaysia for the production of pulp. So in the journey of trying to find new species, suitable species, we need to know what are the species in the water before we can use. So as we carry out the molecular identification, in this case, we actually using RBCL from plastic region and COX-1 from mitochondrial region. And we actually describe 2 new species, which is Thelocladella pangjai and Thelocladella megasporangia. So this is based on the study that we carry out. Okay, another group is Halimenia. It is classified in the family of Helmintheraceae, red algae genus with about 74 species, and they are generally pinkish to reddish brown in color, flattened and rubicous thallus. And this species, it can be a lot of branching, or foliose means a sheet, just like a sheet of paper. Later I'll show you. And then they can be found in intertidal and subtidal habitat attached to rocky bottom, shell, or dead coral. Okay. They are gelatinous thalli. You can, when you touch them, you found that they are like a bit sticky, got gelatin. Okay. Then these are their structure. When you look into microscope, they have filaments, antikinary filaments, and refractive ankylosal medulla. They are stellate cell in inner cortex, and then they have this accessory cell ampulle. Okay, and most of all is this group of species or seaweeds actually had a lot of potential. Would encourage, if for those who are looking into biocompound, you can look into that for your local species. And based on my one-time sampling in Lombok, I found that you're quite rich in terms of the composition of the Helminthia. And in general study have found that they are high in potassium and vitamin B2, and they can use in thickener and fragrance as in seafood soup. This is because they are also very rich in carrageenan. And actually, various study have found that they have anti-cancer and antioxidant compound, and also source of carrageenan. If you ask me if they are source of carrageenan, why these species are not being cultured, I know that I have seen picture of some small number of farmers in Indonesia that actually culture, in Bali, actually culture this Helminthia, but I'm not sure the current stage, whether they are picked up or not. They are good source of carrageenan, but I would say probably the challenging parts is the post-harvest, because this group of seaweeds is easily get rotten very fast compared to Carpophycus and Euchema. So if we can find a way of treating the post-harvest of the usage, probably this is a good group of seaweeds to be explored. So this is the common of the species of that is found recorded in Malaysia. So before that, before we embark this our using molecular approach, their publication have found that there are Helminthia dilatata, Helminthia duraceae, Helminthia foresi, and Helminthia formosa, and Helminthia macroculata. But based on our study, we actually managed to found two new species. One is Helminthia malaysiana and joharensis. And then I think based on our preliminary study in Lombok, you also have this joharensis and malaysianus in your water. Okay, so this is just to show you that how we have carried out morphological study, and how this morphology, it can be very tedious. You need to be specialized, trained in taxonomy in order for you to be able to train and identify. So that's why you will need DNA method for confirmation in terms of species. Just now when I introduce species, I say that for this genus, you will have branching as well as foliage. So this is the sample of Helminthia delivery that actually we consider them as a branching seaweeds. Okay, like for the new species that we have, these are both foliage, so they are like spread like a sheet of paper without any branching. Okay, because we call them foliage type. So these are Helminthia delivery that we look in terms of the thallus, how the construction of the thallus is the branching. In seaweed we call them thallus, and then the color range from dark pink to brown color. The tip are not branches out. Okay, And then, but the highly branched are up to 7 order. So the upper branch up to 7. They can be smooth surface, absent of maculates, margin is highly dented. So at the end, they have a dentate. So you have to look closely, okay? Another species, this is folio. They don't have branching. They are folios thallus. They are dark pink to reddish color, present of sharp, steep margin, minutely dentate, and smooth surface. So especially the folios species, like for example, this is Helminea dilatata. Then you look at Helminea meticulata. Okay, they are quite alike. So it's just that this one is less branch. At the end, they are less dented. Okay, and this is more. Okay, and then for Malaysia, Helminea malaysiana that we carry out. Okay, when we do it, we are not sure and we don't know that it's new species, but based on the molecular data, we found that this is new, and then we start to look into the detail of the morphology, and based on that, we start to identify. Okay, so this is in terms of the using the microscopic examinations on the cells. We look into different aspects, and then as well as also the reproductive structure like tactile sporangial, corpus phyte, how does they look like? Okay, and then so as for Helminea johannessis, we also look into the different structure from the cystocardium plant as well as tactile sporangium plant. Okay, as well as also the cortex cells and corticals, medulla. Okay, on the section. So with that, we also with molecular data as well as support with the morphological features, we name them a new species. And then basically you can see that we have to compare, okay, because the species that we publish new species are folio. So we compare them with existing species, okay, and what are the features that we look into? We look in terms of the thallus size, the blade shape, the blade margin, the surface, the tips, blade thickness, the cortex layer, thickness, shape of outer cortical cell size, as well as shape of inner cortical cells. It's quite tedious. And inner cortical size, refractive ganglion size. And based on that, we get found there are some differences, and these have always to be supported with molecular data, or else you can't name them as a new species for confirmation. Because remember, from the start or beginning of lecture, I say seaweed are plastic. They can change their morphology based on external morphology, external growing condition. So we need to confirm using DNA marker. So with that, we publish the data. So Malaysia, we have delivery. Delivery is complex. You can see that samples from our Southeast Asia and compared with South Africa and Tanzania, they are different. And then we also have different clades. And these are possibly some new species that we have to further to look into to sort it out. Okay. And then Helminea maculata, these are quite and then for Philippines, they publish this is, they say that is a form of the dilatata. And then based on study, we found that the Helminea malaysianus is a new species. And then another one of this is S1, which we use 7 sample. Helminea species A is actually eventually is the Helminea johannessis that we carry out. Okay, so the study that we carry out. Okay, so I would like to introduce another group of seaweeds that we use in order to identify. Okay, can you see any seaweed in the picture that I show you? Any species? Can you see? Okay. Actually,
Not yet.
no. Okay. Actually,
No.
I want to tell you there are seaweed. Okay, this is the kind of seaweeds, a group of seaweeds that you don't notice them, but they are present. If you next time you go to the sea in the rock, you can see them. Okay, they are actually a group of species that we call, okay, we call them crustose brown seaweeds. And these are the how they look like. If you close out, they grow on the rock surface. Even it can grow on plastic straw or steel. So any hard surface in the sea. And this is a group of seaweeds. Okay. So they actually have uses and application, because normally when they grow in the shore, they are always being hit by the wave, harsh environment. They actually develop an effective defense mechanism, and they can be a rich source of bioactive compound. In general, for this group, we call them crustose brown, because they live like a crust, and then this is a type of brown seaweed, so they call it crustose brown. But the type genus in this species is called Rafflesia. They are resistant against grazing and falling activity. They can produce tannin and also repellent against macro seaweeds and microorganisms. So, for example, the ethanol extract of Rafflesia varicosa show activity against some of the microbes, and also the producer of non-toxic antibiotic agent for main-max structure by incorporation to plane. And then another group of crustose brown is Analipus japonicus. Okay, they are actually eaten in Japan, and they have high level of omega-3 and pyrotannins. Okay, with a strong radicals tangenting property. Okay, despite that, this group they are saying to be a crustose species means they do not have erect, they don't have the branching come out. So, but based on molecular analysis, okay, we found that the genus of Analipus is also considered in the family of Rafflesiaceae. But for these Analipus, they are very different like morphological, but based on DNA sequences, they are very similar. So it's quite interesting. Okay, so if for line history of Rafflesia varicosa, the gametophyte and sporophyte, they are the same. But for Analipus, in year 2007, and then we established the family of Rafflesiaceae. And then subsequently, as we want to describe new species, COX-1 is incorporated to support. Okay, so this is to show you the summary of the study. We collected this specimen from Okinawa, which the another phycologist actually named them as Mesospora spineti. So, but the type locality of Mesospora spineti is in Lombok. So with that, that's where how I start to get to know members in UNRAM University, that they host me when I come to look for the Mesospora spineti. And we found that the one that we have in Japan is different, which is Mesospora elongata. Later on, we named them. And then at the same time, we also found that Indio fascicata found in Lombok as well as Malaysia. And then we have another species of only found in Lombok, and we name it after Lombok which is Mesospora lombokkensis sp. So this is how we carry out. So this is example of showing you the three group of the gelatinous light and the filaments. You see, they are growing in the rock. So when we scrape it out, looking under the microscope, and then these are the structure. This is a polylocular. This is unilocular. And the three main group, Mesospora, Difura, and Neoraphsia, they are a bit gelatinous, slimy when you touch them on surface, but when you look into the microscope, they are loosely, this is tightly, then this is when unilock, they are loosely. So in this case, is very difficult for you to distinguish and purely say them. So in this case, that's where we need to incorporate the molecular marker to identify them. So based on that, okay, we compare with existing species like Mesospora sminthi is the type species, and then Mesospora negosensis that is described from the Philippines, and then from that we describe the species from Okinawa, Japan on elongata. We name them new species, and then subsequently Mesospora indificata from Malaysia and Lombok, as well as Lombokkensis that found in Lombok. And we also have another one of Mesospora species D, which we have yet to publish on this. We are looking for more information. So, and then another group is Difura. Difura, is supposed to be formed as a new family. But the thing is, we cannot get the type locality species, and we are still ongoing, okay, the work. So these are some of the work that we look into in terms of the when we establish the new species, okay, first study, and then subsequently we come back. I've started in Japan, then we start to look into Malaysia and Lombok, and that's where we found more interesting new species. And we are, in fact, the tropical species of crustal brown is different from the temperate, okay? The genus that is available is different, okay? So this phylogenetic tree is quite complex, but I will highlight some of the study. And we found that we have this Mesosporaceae, which is actually the species, the filaments are loosely and they are kind of slightly slimy. And then Neoraphsiaceae is slightly slimy, but it's different. And then we also have another group of slimy that's Difura. At the moment, it does not have any family because we need to confirm that the type locality species is actually in America. We have tried a few attempts, but we didn't manage to collect the type locality. We go to the Iberian, the Natural Museum of there also, they cannot locate the samples. Okay. So sometimes there's difficulty when in the old day when they name a species, there's no proper naming. So there's a lot more to work on. But in short, I want to say is that we need molecular marker to identify the species. Okay. So in this case, for this group of algae, we need to sampling of genera previously assigned to Raphsiaceae for molecular study in order to clarify their ambiguous taxonomic position. We need to revise the whole group, establish a new family for Difura, okay? Then wider biogeographic coverage to obtain information regarding the biodiversity. Then gene sequencing at species level for large genera such as Raphsia and Pseudolithoma. And then we have to propose new name for Raphsia expensa from the Western Pacific, because Western Pacific samples is different from the Indo-Pacific at the moment. Okay, so I share with you many methods, but I would want to highlight that the best method for us to use is DNA sequencing. So what are the steps that you have to do if you were to carry out this research? Firstly is you have to collect the sample. When you collect the sample, you have to collect the whole plant, including the whole fast. Okay, seaweed, we don't call them roots, we call them whole fast. Whole fast is the structure that actually attach to any surface where they hold them and grow. So you have to collect the samples, and then you have to, for morphological study, you have to prepare a herbarium samples. If at the same time, if you want to do a formalin preserve also, you can. Okay, 4% formalin in seawater, you can keep them. But the thing is, for formalin, it's easy to check the structure because they won't shrink, but it's a bit carcinogenic, and also the color will be dry off. But I suggest that you keep, at least you must have a burn sample, because nowadays if you publish any publication, most of the reputable journal, they have already come out that you need to have a voucher specimen with the herbarium number, and then you can tell them. And for bioactive study or pharmaceuticals, normally a genus level is sufficient enough, but it's better if you can identify to species level. Beside that, you keep herbarium. You also need keep for sample collection that use for DNA extraction. So in DNA extraction, the samples from the field, you should dry them with silica gel, and you continue to change the silica gel until it's properly dry. Then you can keep for years, a month. But in between, you make sure that you are not introduce moisture, because we are living in tropical region that we have high moisture. So sometimes the samples can be wet. So a lot of time you either keep in a cabinets that actually will maintain there won't be humidity. Okay. For the sample you collect, then you subject it for DNA extraction. DNA extraction, you need to optimize onto the species, the group that you work on. And then you decide on the region that you want to do. You have to decide which region you want to do, and which region is determined by the species, the group that you want to do, or you want to solve which level, species level, or even the strain level or genus level. Okay, once you have amplified the amplicon, the amplicon will be subjected for DNA purification, and then you will send for DNA sequencing. Once you have the sequences, you will go through the bioinformatics. This is the electroprogram that you get for sequencing, and then you will go through the phylogenetic analysis, and based on the data you get from DNA sequencing as well as morphological data, you can conclude the type of the species that you use. Okay, so basically my lecture is until here. Happy to address any question if you have. Okay. Okay. Do you
Thank you. Thank you very much, Prof. Lim. I welcome any questions. Yeah. You are welcome to address the questions in Bahasa also. If you have any questions, please raise your hand.
I have question to Prof. Lim.
Yes.
Okay. Thank you, moderator, for the opportunity. Thank you very much, Prof. Lim, for the very interesting lecture today. My name is Hasriatan Patmi. I'm from Biology Department. My question is how to determine if we have a sample such as Gracilaria sp. If we want to sequence that sample, how the method to sequence that sample using RAPD marker. Thank you very much.
Okay, if you talk about Gracilaria, I actually have quite a lot of experience when talking about Gracilaria. If you want to determine the species, I would suggest you go straight for sequencing rather than RAPD. RAPD is you are comparing between the different species. You want to see whether they are similar or not. But if you do DNA sequencing, is more direct. And then for the primer that you use RBCL and COX-1 for Gracilaria. Then you can determine directly the species that you use. I understood that the cultivations in Indonesia, Gracilaria species that you're use Gracilaria varicosa, which based on my experience, I do not think is Gracilaria varicosa, because varicosa is a temperate species. I do not think it exists in tropical, because in Malaysia, we have collected Gracilaria from whole Malaysia, and we do not encounter the varicosa in our region. Yeah. Am I answering your questions?
Yes, Prof. Thank you very much.
Yeah. Okay, I think I have a few questions from the chat box. Okay, I will answer from Wanda. Is from bagaimana cara menentukan primer yang digunakan untuk analisis gene sequencing dari spesies lumba laut yang ditemukan, dan seberapa pentingnya pemilihan primer ini di dalam analisis yang dilakukan. Okay, very good questions. Okay, firstly is when you collect the seaweeds, based on the morphology, normally you can guess the group, probably genus level. Or if you don't know, you just say red seaweed or brown seaweed. So if you don't know at all, my advice is using RBCL marker, because that will actually lead you to very close and correct answer for both red seaweed, brown seaweeds, and green seaweed. Because RBCL, if in gene bank also, you have quite a sufficient number that allow you to do comparison. Yeah. So, and another one is Siti Zania. Okay, you want to know the identification. We know that the identification process has a very important role to determine the species of seaweed. When seaweed of the same type might have a different morphology due to differences in the environment, does it have a different in the DNA sequences? Okay, there are two things: morphology that are same species but they look differently, is because of the plasticity and the differences. But they are also probability that they are same species but they are different haplotype, means as strain level is lower than species, they are different. So that's why cause, then in that case, you have to use different type of marker if you want to look into strain level. And sometimes strain level, you might have to use like RAFP mitochondria marker. So that is possibility when you talk about strain differentiation that you have to look into. Okay, I hope I answer your questions. Any more questions?
Apakah sudah menjawab? You can respond to Prof. Lim directly if you want. You're welcome. Any more else questions? Prof, I think the Halimania species is very interesting. Previously, I think I found one in Bali, but now it's very hard to find. I think they produce lambda carragina, if I'm not mistaken.
Yeah, correct.
Lambda carragina.
They have a very good, if you are in, because I know Eka, you're interested in pharmaceuticals. Actually, you can look into in your the area that you have a lot of Halimenia is at your tri cultivation area. What is it? The
Gerupuk
gerupuk ekas. Telok ekas you have a lot. You can do the diving over there. They have
We have another question.
Okay, we have from Muhammad Wisam Vira from Pajiwaya University. Okay. About some method that you present, does the best method give the best result or easy to use method? I would say that in the old day, when we had less information, we are exploring different methods. But as we have more study being carried out, so if you are working on the same group of the species that you're working on, then it's good that you follow the data that have most in GenBank. That allow you to do comparison. But if you want to do a specific identification, like strain identification, like probably you know they are same species but different morphotype, then you do a combination of DNA sequencing, determine their correct species, and then you do a more refined, like you go to SSCP or mitochondria, then you see whether they have any genetic differentiation or not. Because DNA sequencing is only based on a single location or region. But if you talk about SSCP or RAPD, they are looking into the whole genomic profile level. Then you will look into. So you need to plan the research question you have. What do you want to solve? Then from there, you decide the best method. Yep. I hope I answer your question.
Yeah, you are welcome to respond lively. I think the students are shy. I'm not sure.
Yeah, you can open the mic and talk.
I mean, in Bahasa Indonesia is Prof. Lim can also respond in Bahasa if you
Boleh.
are
Bahasa
shy
Indonesia.
to. Ya, jika malu pakai bahasa Inggris, bahasa Indonesia gitu. Ada pertanyaan lagi? No more questions?
Got Fitriani raise. Fitriani, you can open your mic and ask question if you want. Fitriani, she raised her hand.
I think she's freezing.
Oh,
Koneksinya. Okay. If we have no more questions, I think we can close this session. And Atmi, or you want to say? Yeah, probably we have a certificate of appreciation. Thank you very much for your lecture today. And um, if the students have any questions, you are free to ask in the group chat, and then we can transfer the question to Prof. Lim, or are the students allowed to contact you directly, Prof. Lim?
Yeah, can.
Okay.
Dr. Eka, thank you for the certificate of appreciation, and also would like to thank you all the students for your attention and listening to my lecture. Thank you. Yeah.
Mohon maaf. Before we close this session, please, for all the participants to on camera, we have photo session. Dimohon untuk kepada seluruh peserta kelas MBKM untuk mengaktifkan kamera. Kita akan melakukan sesi foto. Oke. Oke, untuk slide pertama. 1, 2, 3. Selanjutnya, slide kedua. Dimohon untuk peserta yang belum mengaktifkan kamera untuk segera mengaktifkan kameranya. Baiklah, untuk slide kedua, 1, 2, 3. Untuk slide terakhir, 1, 2, 3. Baik, terima kasih kuliah MBKM hari ini.
Oke, terima kasih semua.
Proses akan dikirim melalui grup MBKM. Terima kasih.
Terima kasih. Bye.